All right. 1 second, let me move this out of the way. All right, hello everyone. I'm Yasser, and for those of you that do know me, I presented the first PACademia. So if you're new here, I would like to say hi to you for the first time. And if you attended for me for the first time, I'd like to say hi to you for the second time. That's a very lame statement, I'm sorry. So I'm going to be, the way today is going to work is that obviously we'll follow the new question-based sessions, but I've heard that a lot of you guys haven't delved into chapter 5 properly yet. So I'm going to try my best to explain everything and then give you guys questions and to test how well you guys have understood my explanation, all right? And we're going to be starting with chapter 5. I'm going to be doing amino acids and peptides, and my colleague Obaida, who's over there, he's going to be doing proteins. And then after that, we're going to move on to chapter 4, which is, I believe, energetics. Energetics, yes. And he's going to be explaining it. So hopefully the session won't take more than 1 hour and a half. Who here has the EMS quiz on like 5 or something? It's now? Has 5 or something. So we'll try our best to finish at least 1 chapter before that period of time, because chapter 5, proteins, is a very beefy chapter. It's like, I think, 50 pages. So we'll try 60? 86. There's no way proteins is 86. It's like 50 pages, all right? So that's still a very huge chapter. So regardless, we're going to try our best to sum it all up. Guys, I really want your attention. We don't have a lot of time. So we're going to start immediately with amino acids. So, as an introduction, I want you guys to, this is very general knowledge. So I want you guys, does anyone here know how many standard amino acids we have? Go ahead. 20? All right, that's perfect, that's correct. And what is the standard structure for an amino acid? Does anyone know what that looks like? Anyone here knows? Go ahead. Yes? Yes. Exactly, that's correct. So that's the standard structure for an amino acid, and it's very important because we see later on is that there are different amino acids, and each one has a different thing that differentiates between each other, right? And as we observe these differences, you'll see how different amino acids interact with each other and give you different bonds, different interactions, and how they stabilize proteins as a whole. And that's the idea of the structure. That's what gives us the third question, which is what is the key difference between each amino acid? Go ahead. Exactly, correct. So, can you give me one? Yes, exactly. Thank you. So let's start with the amino acid structure. So does everyone here know? I'm guessing everyone here probably studied this in high school, which is the structure of an amino acid. Which, as she said, it's just a carbon at the center with an H on top, an NH on an amino group on the side, a carboxyl group on the other side, and an R group at the end. So what is the R group? So the R group fundamentally, it's just you could say it could be anything. It doesn't have a specific structure, but the definitive part of it is that it differs between each amino acid. And there are different types of R groups. Remember when you guys did chapter, I think, 3, which is the chapter 3 or chapter 1, it was the one with the interactions, right? The hydrogen bonds and the hydrophobic interactions and all of that. So different R groups will give you different interactions, and these interactions govern how proteins function. We'll see that in a bit when we get to the big proteins, which my colleague Obaida will go on to. So now we're just starting with the small building blocks, if you guys get what I mean. So which amino acid is the exception to this structure? Anyone here knows? Go ahead. Exactly, correct. What's the difference? Do you know what the difference is then? Do you know what the difference is then? Anyone here knows what the difference is? Go ahead. It doesn't contain a chiral carbon. It does contain a chiral carbon. That's not proline. That's a different amino acid. It is a different amino acid. So what is it? Go ahead. Yes, exactly. So proline is different because we'll see in a bit when we get to the classes of amino acids. But you'll see that proline is different because these two are linked with each other, and that's just like a special feature for it. It doesn't because in the exams they usually ask you a lot about the properties of each amino acid, like which ones have sulfur, which one is not chiral, which ones have an OH group, all that stuff. So all of these are very important details that you guys are going to have to know about these specific amino acids. So now that we've seen the structure, I want you guys to look at it instead of as classes. And these classes obviously are, the amino acids are classified based on what each R group has. So if you have a non-polar R group, which could include examples like if the R group only has a hydrogen atom, or if it only has a CH3, or if it has a CH2 and then a CH and then a CH2 CH3 branching out of it. So all of these are examples of hydrophobic R groups because they don't have, obviously, electronegative atoms. They don't have oxygen, they don't have sulfur, they don't have nitrogen. And yeah, they don't have oxygen, they don't have sulfur, they don't have nitrogen. And these are all what make an R group hydrophilic. So if your R group is just carbon and hydrogen, it's a non-polar amino acid. And this brings us to the opposite, which is a polar amino acid, which is ones that you'll usually find containing sulfur or oxygen or nitrogen. And there is some that contain even whole amide bonds. And this is, and you, as we'll see in a bit. And then we'll have ones that are acidic. So these acidic ones are the ones that contain this bond. So they contain the C double bond O and then an O minus in the physiological pH, which we'll also get to later. So it contains this group in the R group, and then we'll see how that actually governs it, how that kind of like influences interactions. Because sometimes the amino acid will bond using the COH in the R group instead of the one on the side, and this complicates things in a way. So just keep it in mind for now, and we'll get to it later. And then we have basic, which is the opposite of the acidic. It has this instead of this in the R group. So do you guys understand? All right, perfect. So let's start with nonpolar amino acids. So this is actually a screenshot from my own textbook copy last year. And I've ticked the ones that at the time Dr. Wasim was teaching us. So I ticked the ones they told us that we should memorize. And I think these are the same ones for you, aren't they? So it's just glycine, alanine, valine, phenylalanine, and methionine. All right. So the whole idea of nonpolar amino acids is that they're hydrophobic. They do not interact with water. And I think I've taught you guys this before in the last session, that these tend to try to stay away from water. So if you have a big protein, imagine a protein as a ball, right? So if the protein is a ball, then the hydrophobic amino acids are going to be inside that ball. Go ahead. Methionine is a hydrophobic amino acids. Yeah, you don't have, at the time, so for you it's different? All right, so for you it's different, so you guys don't have to know methionine, but this is just, I assume that you guys have to know the same thing. So yeah, that's all. So yeah, what was I saying? Oh yeah, sorry. So these are the ball, so the protein is a ball, and deep inside that ball is the hydrophobic amino acids, which stay away from all the water. And on the outside, you have all the other polar ones. You have the acidic ones, you have the basic ones, the polar ones, and all of that. So these are the non-polar amino acids. And I put a special exclamation mark on glycine because it is, they don't know why I put exclamation mark, actually. Go ahead. I wasn't asked. Yeah, it's okay, Go ahead. You were at the back. Okay, it is slightly hydrophilic, but that's not the highlight that I'm looking for. Go ahead. Yes, that is one highlight. It is non-chiral. And anyone else know what's the biggest part about glycine? Go ahead. Okay, that's the same thing as in not being chiral. So go ahead. Yes, exactly. Glycine is the smallest amino acid. Why? Because it has the smallest R group. It only has an H in place of the R group, right? And because of that, it's mentioned that because of that, you use this small property in order to squeeze things. You can say it facilitates flexibility. It gives you the opportunity to be more flexible, because when you have big molecules all next to each other, it's really hard to change their shape and change their orientation. But if you have small glycine amino acids next to each other, it becomes easier. It provides flexibility, and we'll see that later on, is that this specific amino acid is very important in one of the most important proteins in your body, which is collagen. It dominates collagen. So that's something for my colleague to explain. So this is just something to keep in mind for now. So these are the amino acids here. So we have glycine, you have alanine, which has a CH3. Alanine is also pretty important when it comes to stereoactivity, because it is the smallest chiral one. It's the smallest chiral amino acid, and it's the one actually used as a reference. Some textbooks will mention another amino acid, or will mention something like glyceraldehyde, but we use alanine. We'll use alanine for proteins and for specifically amino acids as a reference for which amino acid is which amino acid like rotates light to the left and to the right. You guys know what chirality leads to, right? Because when something is chiral, it's able to rotate light to a specific direction. Does everyone know what that is? All right, you don't know what that means? All right, basically, when a carbon is surrounded by 4 different groups, meaning in this case, NH3, COO, H, CH3, these are all 4 different groups, right? When you pass light through a solution of this compound, the light rotates in a direction, right? You get what I mean? And because of the fact that this has 4 different groups, you can arrange these groups in 2 different ways, right? And because of this, and depending on the arrangement, the light rotation will be different. So if you arrange it in a specific way, the light can rotate to the left, and we call it L lever rotator, which means like left, rotating to the left. So we call it, for example, L alanine in this case. And if you arrange it in a different way, another way, it'll be rotating light to the right. You get what I mean? So, and that would be called D for dextro. Dextro means right. I think you guys should know this in EMS. Eventually, you're going to be asked about it in the final, that D is dextro and L is levo, rotatory. So the reference for amino acids is alanine, and specifically, all amino acids found inside your body are what? Does anyone know where the rated direction of light? Exactly. So they're levorotatory. So this is important as well, because when you have a lot of amino acids together, they rotate light to the left, and they give you rise to right-handed chains instead of, like, you get what I mean? So it's like the opposite. So, and this is a very important property in many proteins as well later on. So yeah, let's not stay too much on this. So this is valine. This is phenylalanine. It is aromatic. It's the only aromatic one you're expected to memorize. And methionine is actually special because it's one of 2 amino acids that has sulfur. Does anyone know what the other one is? Go ahead. Exactly correct. So the difference between this and cysteine is that here the sulfur is non-terminal. It's not at the end. And because of that, it's not able to be fully polar. You get what I mean? So that's why you put it in the non-polar amino acids. So that's it for non-polar amino acids. So now I have a question. Which of the following amino acids, so all of these have hydrophobic side chains except which one? Go ahead. Yeah. Tyrosine, correct, yes. Tyrosine is in the next class, which is the polar amino acids that we'll see in a bit. Kat, be careful. Oh, good job, good catch. Okay, so then we have now polar amino acids. And I think, can you correct me, are these the only ones you're expected to know? Cysteine, tyrosine, and threonine. And serine, sorry. Yes, all right. So I want to ask you guys now, what type of bonds form in these amino acids when they interact with water? Exactly, hydrogen bonds. So, and why is that? We'll see in a bit. So let's start with serine. So serine has a hydroxyl group, obviously, so it forms hydrogen bonds. Threonine, hydroxyl group, hydrogen bonds. This one has an OH at the end of the phenyl group, so obviously also hydrogen bonds. And here, these ones are different because they don't have hydroxyl groups, they have amide groups instead. And these amide groups are more capable of forming hydrogen bonds in that sense. So we'll see actually how these are related to amino acids that are in the acidic category, because these are amides of those. So yeah. So here, this NH2 is what's capable of forming the hydrogen bonds. And at the end, you have cysteine. And cysteine doesn't have nitrogen, oxygen, or fluorine. So it's not conventional in the sense of forming hydrogen bonds. But it is polar, and it does deionize, and it does form polar interactions with other amino acids. And so they put it here. And I put an exclamation mark on cysteine for a very important reason. Does anyone know why? Anyone know why? Go ahead. Yes, exactly. So cysteine is the other sulfur-containing amino acid, and it is the one capable of forming disulfide bridges. So we'll get to how that works later on, but keep that in mind as well. So yeah. So now, I have another question for you. So enzymes are activated by phosphorylation of which amino acid residue? So this sounds a bit of a mouthful, but I want you guys to keep in mind that whenever we mean phosphorylation, we're asking about where is the OH, because usually you put the phosphate group next to the OH. So this question is basically asking you which amino acid which one of those has an OH in the R group? Does anyone know which one? Go ahead. Yes, exactly. So serine is the correct answer here. Everything else doesn't have an OH in it. So keep that point in mind. So now we're moving on to the acidic amino acids. You guys, there's only 2 of them. You have to know both of them. And the first one is aspartate, and the second one is glutamate. Look at the structures, and I want you guys to think about how do they relate with the ones I mentioned previously, with asparagine and glutamine. So if you know, does anyone know what's the link between them? I want you guys to look at these. So this one is a CH and CO and CH2, CH2, CO. And the previous one is a CH, CO, NH2 and CH, CH, CO, NH2. So what's the relationship between them? Go ahead. Yes, so that means you can get one from the other, right? So what reaction would you have to do to the acidic ones to get these? I'm talking about the R group specifically. How do you go from so that group was acidic, and this one is an amide, right? So you can produce one from each. Yes, yeah, you can be. It's not substitution; it's esterification. You don't have to know this, but it's important later on to know that aspartate you can produce asparagine from aspartate by your body can do that, and you can produce glutamine from glutamate. You're going to take this next semester. This is just for your information. It helps to spot these because these are very tricky sometimes because they put the amides in the question and they ask you to find the number of peptide bonds. So some people think that these R groups are peptide bonds, which is wrong. A lot of people make that mistake in the first quiz. So you have to be mindful of which amide bond is a peptide bond and which one is just part of the R group. So you guys understand what I mean? So these are just amides of the acidic ones. So this is aspartate and glutamate, and all you have to know is that they're acidic and they have an acidic group, and they at physiological pH, which is a pH of 7, they're negative. We'll get to see how physiological pH functions in a bit. So just hold on for a bit, and then we have the basic amino acids. So basic amino acids, there's also only 3. You have to know histidine and lysine, I believe. Is that correct? Yes. You have to know histidine and lysine. So lysine has an NH3 group at the end, which makes it basic, and histidine is weird. It's unconventional in its structure because it doesn't have an NH in that sense. Instead, it has something called an imidazole ring or an imidazole ring, and this ring is what acts as a base. So the problem is, since this one doesn't have an NH in the same way that the others do, it's a weaker base. It's a weaker base, and it's not basic to that extent. So you could say that these ones are like, they're basic at pH of 9, at pH of 10. This one is basic at a pH of maybe 8, 7.6, like something in that range. And this is an advantage. This is an advantage because this amino acid is what's used as a buffer, actually. So you guys know what that means, right? Yes, so it's used as a buffer in the blood because it's a weak base, and therefore we can use it to balance things out, if that makes sense. So what type of bonds do these form with acidic amino acids? Anyone here know? So we have these are basic, and they're charged, right? And acidic ones have also a charge, which is a negative charge. So which bonds we form? Go ahead. Yes, ionic, exactly, it's correct. So it is... Yes, so it is ionic because they're opposite charges, and these interactions are also important in the bigger proteins, in bigger structures, as we'll see later on. So yeah, now I have another question: which amino acid is more likely to be localized on the surface of a globular protein? So anyone here knows? Let's break down the question. What does globular mean? Yes, soluble, exactly. So it means soluble. So that means if you're going to be on the outside with a soluble protein, meaning you have to be what? You have to be hydrophilic, exactly. So which one of these is, the question is asking which one of these is hydrophilic. So which one of these is the answer? Go ahead. Asparagine? You said aspartate. Aspartate, yes, exactly. That's the correct answer. Let's look at the other options. Methionine was what type of amino acids? Nonpolar, exactly. Leucine, glycine, exactly. So this is the only thing left. So the questions, this is the questions from the previous. So hopefully the questions will also be like this in your exam. Now this is the question: which of the following amino acids? Yeah. So all of the following amino acids are chiral carbons except which one? Go ahead, Omar. Correct, glycine. Every other amino acid, there's 19 of them that are chiral. Only one is achiral, which is glycine. Which amino acid doesn't have a primary amine? Go ahead. Yes, we mentioned this. It's proline. All the other ones have a primary amine. So yes. Okay, now moving on to the next part. This part is it's not very significant, but it's important to know that amino acids don't have to be part of proteins to serve a function. The amino acid itself can serve a function without joining or doing any peptide bonds or anything like that. So, and these are the examples. Some of the neurotransmitters in your body, like GABA, for example, they're derivatives of amino acids. GABA specifically is a derivative from glutamates. And even the amino acids themselves, without any chemical modification, can act as neurotransmitters or as biologically active molecules, like glycine and glutamate. They both can act as neurotransmitters. And you also have hormones like melatonin. Does anyone here know what melatonin is for? Go. Exactly. So it's for sleeping. So if you have insomnia, for example, they'll give you melatonin pills, and these are derivatives from tryptophan. Yes, and these are derivatives from tryptophan, which is the the hydrophobic one we mentioned in the beginning. And then you also have precursors. So amino acids can be precursors of molecules such as chlorophyll. It's not a very important example. You can just keep that in the back of your mind. And then this is important. So you have amino acids that are metabolic intermediates. Now, these serve a function, but they're not, you can't pull them out of their function. You know what I mean? Like they're part of metabolic pathways, and you'll see that later on in the next semester. They're part of metabolic pathways, and they're stuck in there, and so you can't pull it out and use it on its own. Instead, it's just there as an intermediate. So, for example, all of these are modified amino acids, specifically arginine. Arginine, we already mentioned, was a basic amino acid, right? So arginine, and then you also have ornithine and citrulline. These are all amino acids that are in metabolic pathways. So they're very important for example, these three are mentioned in the process of producing urea, in the urea cycle. You guys are going to take that next semester. The urea cycle is where your body just breaks down like the toxic compound urea into something that can be, breaks down like excess amino acids into urea, and these are intermediates. So just keep that in mind. So do you guys understand everything I said here? All right, perfect. So now this is the part that I think either Dr. Samar or Dr. Ahmed emphasized on, right? So they said that you guys have to be really well versed in the amino acid ionization. So this is a bit of a tricky concept. I'm going to be asking you questions after explaining, but I want you guys to like focus with me, all right? So what is physiological pH? What's the pH that's found inside your body? 7.5, 7.4, exactly. So this is the pH inside your body. And at that pH, all amino acids have a neutral charge. They're neutral charge, which means charge of 0. But within the amino acids, you have charges that cancel each other out. So the amino group in the amino acid at the pH of 7, it has a positive charge. It's protonated. While the carboxyl group is unprotonated. It has no proton. So here you have a minus and here you have a plus. So what does that give you? A neutral charge, no charge, right? And that's the concept of a Zwitter ion. You guys understand that? So let's say I don't want the structure. Let's say I want to put the amino acid in an acidic medium. So when I put it in an acidic medium, what am I adding? Exactly, adding protons. And protons are acid basically, right? What reacts with an acid? What reacts with an acid? Say it louder. Exactly. So which one of these is the basic group? CO is the basic group. It's not CO, it's NH2. NH2 is the basic group. And what happens is that it binds with it, like the NH2 binds to it and bonds to it and gives you a positive charge. So it stays like this, and that's what happens when you add acids. When you add acid, you're adding it to the basic group, and you're getting a positive charge. And the more acid you add, the more these protons are being accepted, and therefore, at some point, all the H plus has been already accepted by NH3. So what happens is that the H plus goes to which group? It goes to the COO group. Exactly. And this one gets protonated. And that's why when you put a lot of acid, or when you put an amino acid in an acidic medium, you get a positive charge, because this charge has been neutralized by the H plus that the COO accepted. Do you guys understand what I mean? So this happens on one end. On the other end, I am removing acid. I'm removing H plus. And obviously, the first group that will remove it will be the COO, because it's already an acid. It doesn't need H plus. So that's why we move here. And then at higher pHs, since the NH2 is a basic group, it'll hold on to the H plus for longer. And at some point, you remove enough acid, or you neutralize the acid to a point that the NH2 cannot hold on to this H plus and then lets it go. So what happens here is that we have a COO and an NH2. So this here has a charge of -1. You guys understand what I mean? So it's a weird concept, because all you have to know is just for example, when I put a certain amino acid in a pH of 2, what does that amino acid have a charge of? What is its charge? Yes, exactly. It'll be positive in the case of basically be positive. And if I put it in a basic medium, what charge will it have? -1, exactly. So this is the basic idea, and this idea does apply to the polar and the non-polar amino acids, because they don't have charged R groups, right? So the only thing that's making the charge or giving anything a charge is these two groups, right? So I have one example, which is glycine. So this is you don't have to know this really well. This is just for demonstration. Is that you'll see here is that at pH 2.3, you have the first pK. You guys obviously know what pK is, right? And you guys know what it means mathematically, but does anyone know what it means in more of a theoretical standpoint? Do you know what it means? Correct. Correct. So PK is a specific pH at which a certain group, half of the groups are protonated and the other half are unprotonated. So that's basically what it means. So at a certain pK, you'll see so at this pK, which is, sorry. So at this pK, which is 2.3, half of the COO groups will be protonated and the other half will not be protonated. And at this pKa, half of the NH2 groups will be protonated and the other half will remain unprotonated. So this is what this curve stands for. This is just knowledge you guys have to understand, because I feel like when I was in foundation, I was confused. Why is the pKa of COO here and why is it here? Why is this not here and why is this not there? And I tried my best to understand it, and this is how I understood it, is because at low pHs, the first thing that will want to get rid of the hydrogen is the COO, because it's the acidic one. And then you have to raise it up to a higher pH and pull out more hydrogen, pull out more protons in order to unprotonate the NH2. So that's why the pKa of the COO is here and NH2 is here. Does anyone understand? All right, perfect. Does anyone have any doubts? All right. So this is just further illustration. This is just where you have an acidic or a basic R group, and it complicates things because now you have one more charged group to worry about. So you don't have to worry about this a lot. You guys just have to know that you at a very high pH, you'll have a chart at a very high pH for example, glutamate. Glutamate only has one amino group, right? So at a very and it has only one amino group, and yeah, it has one amino group and two carboxyl groups, right? So at a very high pH, everything will be unprotonated. There is no protons. So when NH2 is unprotonated, does it have a charge? Like when COOH is unprotonated, does it have a charge? Yes, it has a charge of what? So, and if I have two groups in that one molecule, how much charge is that in a total molecule? Exactly. So this is what you guys have to know, is that glutamate at a very high pH has a charge of -2, and at a very low pH has a charge of +1. Another example could be lysine. Lysine is what? Is it an acidic amino acid or a basic amino acid? Exactly. So that means it has two NH2 groups, right? And if I protonate them both at a very low pH, what will the charge of the molecule be? Exactly. And if I put them at a very high pH, what will their charge be? At a very high pH, exactly, -1, because there's only one COO. So think of it that way. So that's how it helps to draw everything and see how everything falls into place. And knowing, COOH, when you have two of them, they both are unprotonated in high pH. So it's a -2. And when you have, for example, lysine, which has two NH2s, when they're both protonated at a low pH, you have a +2. So you guys get what I mean? All right, perfect. So now I have a question. So at physiological pH, which of the following matches the protonation status of the amine group and the carboxyl group respectively? So what was physiological pH? 7. Yeah, 7.5. So basically neutral, right? So the amine group, what will it be neutral? Will it be an NH2 or an NH3+? Exactly. So that means it will be protonated or unprotonated. Exactly. So this is wrong and this is wrong, right? And then you have the carboxyl group, COOH, at a physiological pH. Will it be protonated or unprotonated? Exactly. And so that means this is wrong and this is wrong. So this and this and this are wrong, so this is the only correct option. You guys understand? All right, good job. You guys are catching the concept really well, because this took me like days back in foundation to like properly like latch onto. I only have a few. This is the whole concept. So which amino acids have a positive charge on the R group at pH 1? Positive charge on the R group at pH 1. So keep in mind, when I'm talking about pH 1, I'm putting a lot of H plus in it. So what's my concern? Is it the COO or the NH2? Exactly, NH2. So I want someone to answer this individually. Go ahead. Aspartate and glutamate. At pH 1, a positive charge. Okay, I think, yeah, a positive charge at pH 1. Aspartate and glutamate, they're both acidic amino acids. So at pH 1, you'd have, yeah. At pH 1, yeah, you have a positive one. I think it's scary enough to my head. 1 second. Aspartate. I'm not imagining things properly. So you have two, yeah, at pH 1, you'll have two COO and two COOH, and they'll both be protonated at pH 1. And the NH2, yeah, that'll be positive charge of +1. Yeah, exactly. Oh, they're asking on the R group. I'm sorry. Yeah, no, on the R group is a negative charge. I'm sorry. Yeah, I have to read it properly. A positive charge on the R group. It should have a positive. The Pardon? Yes, correct. The basic one. So it's not aspartate or glutamate because I remember it being that. And so basically here what they're asking is that which ones are the basic ones because they're the ones that have the NH2, which gets protonated at a low pH. So which one is the answer? Go ahead. Yes, correct. So yeah, I think this was a previous question, and I think this was a previous question, and it's important to note that you have to read the question. I didn't read the question here personally. I'm sorry for that. So, and you have a charge, guys. So you have the charge on the R group. So you have to basically just keep mind on what's the R group there. So if you look at glycine and alanine, there's no charged R group, so there's no charge. It's wrong. And you look at arginine and asparagine, they don't have a charged R group either, so it's wrong. Look at aspartate and glutamate, they have a COO, and a low pH will just be COOH, so it'll be uncharged. So lysine and histidine have the NH3 or the basic groups, which get protonated at a low pH, and that's why they have a positive charge. All right? Right. Go ahead. The basic group, okay, it has an imazidol ring, and yes, there is a secondary amine in it, and I'm pretty sure it's one. I think it's the second carbon from the. If I go back to this part, is that I think it's this one that is the one that donates that accepts the H plus. So this is the part. But what I was emphasizing on is that since it's a whole ring and there's resonance inside the ring, it's a weaker acceptance, and that's why its pK is lower. You get what I mean? So it's not a conventional NH3 plus like the others. I wanted to emphasize on that. All right? Yes. So there's not much left. All that's part left is just the different amino acid reactions and the different peptides. So there's only two reactions you guys have to be like very aware of is the peptide bond formation. Everyone knows how this happens. The COH on one amino acid and the NH2 on the other amino acid, they form an amide bond, the peptide bond, and then release water. So now I have a question. So look at this molecule. This is the type of question that comes in the exams. They give you a large molecule in skeletal formula, and they ask you how many peptide bonds are in this molecule. So can anyone tell me how many peptide bonds are in this one? Does anyone know? I want new hands. I want new hands. You're not a new hand at all. I want new hands. Anyone there at the back? You guys have any idea? Go ahead. 3? All right, let's see. So there's 1 here. There's 2 here. There's 3 here. Okay, it's not 3. There's more. Go ahead. Exactly, correct. So there's 1, 2, 3, 4. So this is what I mean when I get confused, because when you look at this large molecule, there's so many NH and so many COs that you might undercount or overcount the number of amino acids. So just keep track with the main chain. So here you have CO, NH. Done. That's 1. CO, NH. That's 2. CO, NH. That's 3. CO, NH. That's 4. This one is just a R group. This is, I believe, aspartate. If I'm not wrong, yes, I believe aspartate. So don't be careful not to count this or double count this bond. You get what I mean? So everyone understand? All right. And the last reaction is cysteine formation, which I believe you mentioned earlier, right? The formation of disulfide bridges, right? Yes. So this disulfide bridge formation is an oxidation reaction. Why? Because we're removing an H from both cysteines. And when they bond, a covalent bond is formed between the S and the S, and that forms a molecule called cysteine. They're both called cysteine. The only thing that's different is the spelling. This one just has an I instead of an EI in it. So you guys understand? All right. And then you have the last part of my part, which is the important peptides. You guys don't have to know a lot about this. You guys just have to know, I believe, the structure of glutathione and the fact that it has a unique structure. And you just have to know the functions of oxytocin, vasopressin, and ANP, right? So let's start with glutathione. Did anyone know let me just hide this. Did anyone know what the special feature of glutathione is? Yes, it is a gamma peptide bond, yes. So it's important to just look at it. So here we have this is the first peptide or the first amino acid, and it's glycine. And then we look to the next one. This one has an SH at the end. Okay, it's cysteine. Now let's look to the third one. This is the amino acid, and then we have a CH2, and then we have another CH2. So this is weird. We expect after this to have an NH, right? That's because this is the main chain of the amino acid, which is, I believe, in this case, aspartate. No, sorry, glutamate. Glutamate. So, which I believe in this case is glutamate. And this is the R group, and it has CH2, CH2, and the COO in this case of the R group is bonding with cysteine instead of the main chain. And that forms a gamma peptide bond. You guys know how the naming works, right? So this is an alpha peptide bond because the carbon straight after the CH2 is the one that's bonding with straight after the main chain. So this is an alpha, and here you have one. So that's the main one, and you have this is alpha, this is beta, this is gamma. So the gamma and then the carboxyl after it is what's bonding. So that's why it's called the gamma peptide bond. And this is just an antioxidant, so it's very important. It's actually very important in a kind of antioxidant before it comes to blood, because sometimes the hemoglobin in your blood goes wrong and it forms a different compound called methemoglobin or something like that, and that's a very toxic compound, so you have to break it down. And then you have oxytocin, which is just for uterine contractions, especially during birth. And then you have vasopressin, which is important for raising the blood pressure. It causes systemic vasoconstriction. Just keep those in mind. So this raises blood pressure. This is for uterine contractions, and it's very important to notice that these structures are very, very similar. They both have a disulfide bridges, and the amino acid sequence is almost identical. The only difference between them is this isoleucine and this phenylalanine. They're different, and the leucine and the arginine here are different. So it's very important to see that this just demonstrates that even as one change in one amino acid could change the function of your whole molecule, because there's a huge difference between being responsible for contracting the uterus and causing like systemic vasoconstriction, right? So this is just for demonstration purposes. And then you have ANP, which just lowers the blood pressure. That's all you guys have to know for this part, and I hope you guys enjoy my part. I'm passing the floor to my colleague Obaida. Oh, there was a question. This is my last question, I believe. Of the following peptides, which one are non-peptides? Does anyone know what a non-peptide is? Yes, 9. So 9. So we already know that glutathione only has 3, right? So which one do you guys think is the correct answer? ADH? Oh yeah, it was ADH. Yeah, it was ADH. Yeah, sorry, it was ADH. ADH oxytocin, correct. Good job. Yes. Can you pass the thing to her? It's her. Here you go. Yes.
Yasser, حطها على الداخل, or is it going to be too loud? Like here or? تعال ولا where's my Yasser? Where's my PowerPoint? Where's my PowerPoint? Oh. Okay. Chapter 5 protein. Okay. يلا. السلام عليكم. First of all, the slides were done by Lujane Samar, so credits to her. I didn't have enough time to make my own slides. I apologize for that. Yasser, when Yasser? You didn't put the slides that I told you about. I did not. Okay. So start from here. First of all, the first thing that you have when you start 5.3 is a function of proteins. This is just basically a memorizing part of the book. You just have to memorize each function and an example of it. That's the only type of questions they could ask you from this. Most of the time they can ask you, for example, in which of these would you find actin and myosin? Which of these functions? Movement, صحيح. وين حسن؟ ياسر تعال وزع حلويات. ما أعرف. Who answered?
Well, I know these, but I want to use up the other one.
Okay. Who answered? Okay. multifunctional proteins, also called moonlighting proteins. These proteins have more than one function. For example, GAPDH, which is a dehydrogenase, its main function is to oxidize reactions, but it also helps in DNA repair and DNA synthesis. And the classification of proteins works based on a few things. First of all, the sequence and the 3D shape. It's first divided into families and then superfamilies. Superfamilies are like big families such as globins, which include hemoglobin, myoglobin, and neuroglobin. And it can be classified by shape and water solubility into globular and fibrous. For PowerPoint. And then composition, which is simple and conjugated. Simple proteins are proteins that can function on their own. Conjugated proteins need something called a prosthetic group, which is required for the protein to function properly. The protein with the functional group or the prosthetic group is called a holoprotein, and without it is called an apoprotein. And now, this is very important, the structures. First of all, we start from the primary structures. It's important to also associate the bond type with the structure. So for the primary structure, it's just basically the peptide line that comes out of the ribosome, أول ما يطلع. It's just one amino acid after the other, and it's stabilized by peptide bonds. And the arrangement of amino acid is what gives the information for the protein to fold into a 3D shape later on. Homologous proteins are proteins that have a similar structure and function across different organisms. It's thought to be arise from the same ancestral gene. And now you can have mutation. As I said, the order determines the folding. So the mutations, they could affect the way the protein works or not, and it depends mainly on two things: the function. For example, the invariant residues. The invariant they cannot be replaced. So if you change that protein with another one, it's gonna destroy the function of the protein and cause problems. While the variable ones, they're mainly structural proteins, they're not important for function. So changing these proteins would usually not result in a big problem. And by the amino acid class, for example, if the amino acid is chemically different, such as switching an acid with a base or a polar with a non-polar amino acid, it's gonna cause polarity changes that can affect. If the change does affect the protein function, it's called a non-conservative change. If the change is from a similar amino acid class or similar amino acid family, it's gonna create something called a conservative change, where there is not much different in the protein function. One example of molecular disease is the sickle cell anemia. You have glutamate is switched to valine. Can anyone tell me the class of glutamate and valine? صحيح. منو لازم الحلويات؟ Exactly. So what type of change is this? Does anyone know? Just mentioned them the slide before. صحيح. Okay. Secondary structure. As soon as it gets out of the ribosome, it starts folding. The first step in folding is folding into a secondary structure. Not many proteins have the final structure as secondary, but usually the proteins whose main function is structure and it doesn't have a biological function, it would be left at the secondary structure. The most important bond in the secondary structure is the hydrogen bond. So they're considered weak individually, hydrogen bonds. You probably know them from chemistry that they're the strongest type of bonds. But in biochem, they're considered the weakest as there are stronger bonds. But together, when there's a lot of hydrogen bonds throughout the structure, they're going to become strong. So the hydrogen bond happens between the amino and the carboxyl group. As you know, the hydrogen bond is between an H and then either nitrogen, oxygen, or fluoride. The hydrogen bond happens in amino acids that have a distance of 4 between them, and this is 1, 2, 3, 4, and then on the fifth one you have a hydrogen bond. Now there are two types of secondary structures. There's the alpha helixes, which is just one polypeptide that's wrapping around. That's the one that I talked about. It has 4 amino acids between each hydrogen bond, and there are B sheets, beta sheets. Now, with the alpha helixes, there are a few amino acids that cannot work with the alpha helix. One example is proline, as you know, because proline, the R group is linked to the amino, therefore it cannot continue the chain and it would end with it. And glycine, because it is too small, highly charged amino acids and bulk R groups, all of these they destroy the helix. Now, for beta sheets, there are two types: parallel and anti-parallel. The anti-parallel ones, the amino group and the carboxyl group are put on the same level, and the anti-parallel, they're on separate levels. One is a mirror image, the anti-parallel is a mirror image, while the parallel is not a mirror image. And the bonds in the parallel one is weaker. Okay, now super secondary structure, this is between the secondary and tertiary structures. It's basically a mixture of more than one type of secondary structure. And these are mainly stabilized by hydrophobic interactions. These are very important, by the way. You might get asked about them. You can tell by the shape B, and then you have a helix, so that's an A, and then a straight line, that's B. You have B mander, B barrel, Greek key, and AA. Okay, so if I were to ask you between B, C, and D, which one is the weakest? And why? And why? صحيح, because they're all facing the same way, it makes them parallel. As you can see, in the anti-parallel, it's opposite. So the C terminal is opposite to the N terminal, while the C terminal is opposite to the N terminal. In the parallel, C with C and N with N. And as you can see, the arrow is where the C terminal is. So all of the C terminals are on the same side, making it parallel, which is the weakest type of B sheets. Now, tertiary structure. This is when the protein starts assuming a 3D shape. And as I said before, the information of the folding is always contained in the sequence of polypeptides. Now, the tertiary structure is important because it brings far away amino acids together to give function to the protein. And globular proteins are compact because of this. They're not spread out. And for example, in transmembrane proteins, the side chains which have non-polar amino acids would move to the sides, so the protein itself can embed into the lipid membrane. Now, domains are the functional parts of tertiary amino acids. Each domain has around 200 amino acids or more amino acids. It can have repetitive domains, such as here. Each F is a domain. So F1 is a domain, F2 is a domain, and F3 is a domain. As they become repetitive, they're called mosaic, because it's just a repetition of multiple domains. This is fibronectin, and it has mainly three domains, F1, F2, F3. And as I said, everything is coded by the sequence of polypeptides. Now, this is the example of domains. We had the question coming from this in the final. It was, what was the function of leucine zipper, DNA binding? And I recommend memorizing them. There isn't much to explain. It's just the way it looks and what its function is. Now, this is important. The interactions between side chains of amino acids that help stabilize them. طبعا here, the way they make you get mistakes in the question is by using multiple different names. يعني covalent and disulfide are both the same thing. Ionic, electrostatic, salt bridge. They might use like a different word in each question to make you doubt yourself, but you have to like know these pretty well. Disulfide bond, it's when two sulfides cross together, two cysteine residues, they're oxidized and they get a bond between them. The ionic, electrostatic, or salt bridge is when a negatively charged amino acid group with a positive charged amino acid group are opposed to each other and they have an attraction. The hydrogen bonds are the same as the secondary structures, which is when the carboxyl group and the amino group of two separate chains are connected together. And then you have the hydrophobic interactions, which is just basically for the non-polar amino acids, London dispersion forces or dipole-dipole forces, you might know them from chemistry, and the hydration, which is when a water molecule is placed in between two other molecules. And now quaternary structure. So quaternary structure is more than one polypeptide. So you have multiple polypeptides. And the polypeptides could be either tertiary or secondary. The functional unit of the quaternary structure is called the subunit. Each single polypeptide is its own subunit. Now, طبعا, last year we got a very hard question coming from this. لحد الآن almost nobody understands the question fully. It was asking about oligomers, protomers. So protomers, the protomer is just one single amino acid folded into a domain, while the oligomer is when more than one amino acid's multiple domains are joined together to form the quaternary structure. And the quaternary structure is mainly stabilized by hydrophobic interactions. And there's also something called allostery in quaternary structure, is when basically one domain, the fact that if it's bound to a ligand or not, affects the other domains. For example, in oxygen, when one oxygen binds to a single hemoglobin, single heme group, it activates the other heme groups, so they get a higher affinity and would bind to oxygen at a faster rate. Denaturation, هاي مهمة. Denaturation is as I told you, as you fold proteins, they gain function. Denaturation is the opposite, is when you unfold proteins and they lose their function. And they could be unfolded to any point, down to tertiary, down to secondary, down to primary. But the polypeptide bonds are not broken during denaturation. That's called hydrolysis. Okay. Now this is just basically what could cause denaturation. You have strong acid or bases that can affect hydrogen interaction, changes in temperature, hydrophobic and hydrogen interactions, organic solvents, polar and non-polar reducing agents, disrupt sulfide bonds and hydrophobic interaction. They gave you an example of reducing agents in the book, which is beta-mercaptoethanol. Does anyone know what it reduces? It reduces disulfide bonds and also urea. It does the same thing. It reduces. So if I tell you that the beta-mercaptoethanol reduces disulfide bonds, which structure of proteins would it mostly affect? Primary, secondary, tertiary, quaternary? Tertiary. صحيح. Salts, they disrupt the ionic interactions, but it's also mentioned in the book that they cause the protein to precipitate. Do you all know what precipitation means? Precipitation, as I told you, the globular proteins, as Yasser said, globular proteins are soluble in water, صحيح? Precipitation is when you add something to that water and make the protein solidify, come out. And of course, if it's not solved in the water, it's going to lose its function. That's what the salt can do. And then the mechanical stress is just generally a 3D shape of a protein. If it's changed anyway by mechanical stress, it's going to lose function. Folding problem. So long polypeptide chains and huge proteins, the folding process for them is hard and could take time. So that's why they're sorted into something called molten globules, which is just basically half folded proteins. For example, if the final structure is tertiary, it could be stopped at secondary, and when it needs to be activated, it's going to need to be continued the folding to get the tertiary structure. Molecular chaperones. These they do the folding for the proteins. And if I were to ask you, what determines the folding structure of the proteins, what would the answer be? صحيح. Amino acid sequence. These, they just fold it. They do not get their instructions from the instructions are already within the same amino acid. So there are 4 types: ribosome associated, HSP70, HSP90, and chaperones. They also, their function is not only just fold proteins, they can also prevent denaturation, and they prepare proteins to be released into this outside the cell. Okay, HSP70s. I recommend reading this from the book. and cross-referencing it with exactly what the teacher said, because there is so much in the book and you don't know what exactly is going to be included or excluded from the exam. HSP 70, it's the one that firstly folds the protein, the starter, and then you have, it's not mentioned here, but HSP 90s, they do the latter folding afterwards. HSP 90s, you get two, they work as a dimer, so two HSP 90s come together and they form a V shape. The HSP 70 has co-chaperones which assist in its work, which are HSP 40 and HSP 70. I know there's so many numbers, so much HSPs, but yani, try to read over it in the book more than once. Chaperones, there are two types, type 1 and type 2. Type 1 is a seven-membered ring, and type 2 is an 8-membered ring. Type 1 needs a lid, co-chaperone. Type 2 has the built-in lid in it. Yani, I recommend reading these details because they asked us last year about the structure of group 2 chaperones, and nobody read that from the book. ما حد جاب العلامة. And also, it was mentioned here, ribosome-associated chaperones. These actually don't really fold the protein. They just prevent it from folding as it is released from the ribosome until the entire structure is released. Okay. Which molecular chaperone binds, stabilizes, and promotes the folding of nascent proteins? صحيح. طبعاً. وين ياسر؟ Now the classes and subclasses of proteins. We have the fibrous protein, as Yasser told you, these are the water insoluble ones, and their main function is to hold the structure. For example, the keratin you have in your skin and hair, it's a double helix structure forming hair, wool, and feather. Fibroin, which is similar to keratin, forms silk and spider web, and it's beta sheets. The only function of these two is structure. There isn't any biological function to them. Collagen. Collagen is formed by triple helices. Be careful, هنا مكتوبة it's helices and not helix. It's not the alpha helix, and it's made from 3 polypeptide chains. So if it's made from 3 polypeptide chains, what structure is it? Quaternary. Glycine and proline constitute up over 60% of the collagen residue, and increased in cross-linking مش مكتوبة هون. As you get older, the cross-linking increases between the triple helices, and as the cross-linking increases, the collagen becomes more brittle. Because, as I said, quaternary structure is when you have more than one polypeptide together. هون عندنا we have 3 polypeptides. Tertiary is when it's folded into a 3D shape. Now you have globular proteins, which are the water-soluble ones. These have lots of examples, and they carry a biological function, such as hemoglobin, myoglobin. They both store oxygen. Enzymes, they catalyze reactions. Peptide hormones, they are signals. And immunoglobins, which are defense bodies. You're going to study about them in biology term 2. I'm going to talk about myoglobin and hemoglobin. طبعا they asked us some questions about these, and they were a bit hard. So myoglobin, it's a globin with a single protein with 8 alpha helices. So if I were to ask you, what structure is it? It's super secondary because it has 8 alpha helices. It is folded into 3D shape, but they did not mention any of the domain of the 3D shape. They just said 8 alpha helices. Most of the people last year فكروها إنه it's a quaternary structure because they said 8 alpha helices, but it's just a single polypeptide chain. Its main function is to store oxygen, and it can store one piece of oxygen. It is in the cardiac muscles and the skeletal muscles, which are muscles in your body, and they store oxygen for later on use. Now, hemoglobin has 4 subunits. Each one binds the oxygen group. You have 2 alpha and 2 beta subunits. So I don't know the difference between the two of them. It's not mentioned anywhere. And you have, for example, the embryonic and fetal hemoglobin. They have a higher oxygen affinity. Some hemoglobin chains have a delta chain instead of a beta chain, HbA2. It's just extra information. And if I were to ask hemoglobin, what structure is it? quaternary. Then what bonds is it stabilized by? صحيح. ها؟ نظرات. Okay. And for hemoglobin طبعا لحظة عشان ما تغشوا. What is the prosthetic group in hemoglobin? صحيح. Okay. Now this is the heme group. Basically, it's an iron with 6 bonds connected to it. 4 of the bonds are just the nitrogen around it. The function of the nitrogen is just stabilize the iron when it binds to an oxygen and unbinds from the oxygen. And there are 2 histidines also bound to the iron. One is the proximal histidine, the closer one. Proximal means closer. And that one is just for stability, and it binds directly to the heme group, while the other one, the distal histidine, binds to the oxygen, and it's the further one. Both are similar. They're very similar, but they're not exactly the same. It's not that myoglobin is one of the subunits of hemoglobin. They have some differences. Hemoglobin has, as I said before, it has allostery. As one oxygen binds, the dimers would shift by 15%. It's not that important, but what happens, it increases the affinity of the next subunit to bind to oxygen. That's why the binding of the first oxygen would speed up the binding of the second oxygen and third and fourth. The deoxyhemoglobin is called tout or T, and the oxyhemoglobin is relaxed because, as you know, proteins, they're considered unstable because they need to be reactive. But in the relaxed state, they're more stable and less active. Now this is the curve explaining basically the speed of oxygen binding. And the myoglobin, it has a hyperbolic curve, which basically, as the pressure increases, the O2 saturation gradually increases. While the sigmoidal curve, this is the binding of the first protein, and then it speeds up the binding of the second and third and fourth protein, and then it's fully bound. Now, the main function of this is that so the oxygen has a higher affinity for O2 and can transport it easier from the lungs to the muscles. And this function is also called cooperative binding, where one subunit helps the other subunit to bind to oxygen. Now, there are 2 main things that affect the O2 affinity. The first one is the Bohr effect. So at higher pH, you're going to have higher affinity to oxygen. At lower pH, you're going to have lower affinity to oxygen. So if I were to ask you, what would the pH around the lungs be? Would it be high or low? صحيح. And in the muscles? Correct. Why is that? صحيح. Lungs, the entire point of them is to load the hemoglobin with oxygen. The muscle, it's going to need the oxygen, so it's going to need the blood itself is going to need higher affinity for oxygen near the lungs and lower affinity of oxygen near the muscles, so it's easier for it to release the oxygen. This is from the biology. I don't think you studied this yet. It explains why around the muscles it's more acidic, around the lungs it's more basic. But I don't think this is included for you guys. Also, the same thing with temperature. As I told you before, temperature can cause denaturation. It can affect the hydrophobic and hydrophilic bonds. So if temperature increases, O2's saturability would decrease. And then you have this BPG, 2,3-bisphosphoglycerate. This stabilizes the deoxy hemoglobin. Yani the function of it is when the blood gives donates the oxygen to the muscles, the BPG would bind to the blood and make it more stable. And you'd of course expect to find more BPG around the skeletal muscles. The function of BPG هاي لسه قلتها stabilize deoxyhemoglobin, which is طبعا ياسر الله يستر عليه الولد. He was supposed to remove the highlighting بس وين حسون؟ Thank you. ياسر تعال تعال. This is so empty. I didn't upload the latest version of the PowerPoint that I sent. مم. لا يعني
Okay, I hope this is good. I'll be talking about energy. So energy, what's energy? How is energy transferred? Basically, thermodynamics you took from AP chemistry. Okay. So first I need to define some terms for from 4.1, which I understand is excluded, but it will make understanding 4.2 and 4.3 easier. My references will be the chemistry book right there, the chemistry book that you took, and YouTube. So what's energy? Can someone define it? The capacity to do work. Okay. Are there types of energies? Yes. Can someone tell me some examples? Written on the board. Work and heat. More. Work is not a type of energy. Kinetic, chemical. You have electrical, electromagnetic. So there are types of, as I explained, there are types of energy, and there's also quality. There's high quality energy and there's low quality energy. So in your book, high quality energy will be electromagnetic, electrical, and chemical, while the low quality will be the thermal. So continue. The energy is given by this equation: delta E equal Q plus W. Does anyone know what Q and W means? Okay, so Q is heat and W is work. It's written on the board, please. When delta H or enthalpy, which is equal to delta E or energy in biological situations, when it's greater than zero, the reaction is endothermic. That means that the system is absorbing energy. When delta H is less than zero or negative, that means that the system is giving off energy, which means that's exothermic. Can someone define work? Okay. I have a quick question. Did anyone revise chapter 4? Did you have time to catch up to chapter 4? Okay. No. Little bit. Okay. So work is direct energy change resulting from a process. This is defined from that book. From your book, it's defined as focusing of energy to move a process or a motion. Okay, for example, as we took in physics, work is distance times force. There's also work in thermodynamics, which is change in pressure and volume. Okay, so an example of work is a ball falling from a floor, from the roof, for example, or pushing a table. Okay, so here's an example question. A scientist measured the amount of energy released from ATP to be 7 kilojoules per mole to pump sodium outside of the cell. You took this in cell midterm, correct? What type of chemical energy what type of energy is released? 1, 2, 3, or 4? 1. Anyone else? 1. Who answered? Right? Yasser. تعال وصلها. Okay, so it's chemical energy. It's high quality because, as I explained, electroradiation and chemical energies are all high quality, and work is done because a particle is moved from inside the cell to outside. Move on to the systems. You all took this in high school, I believe. So do you know the type of systems? Can someone tell me what's the first one? Open system. What does open system specialize in? What's so special about it? Interacts with the environment. So it releases energy and mass. Yes. Now what's the second one? A closed system. What's different from an open and a closed one? Correct. And what's isolated? This is the last one. It's not in your book. Isolated. Correct. Isolated means that neither matter nor energy can move from the system and surroundings. واحد الولد شبه هذا Entropy. Have you ever heard of entropy before? Yes. Where did you hear it? Can someone define it? Disorder. Okay, so entropy is the change in disorder of a system. That's also defined in your book. For example, putting a cup of hot coffee on a table, what will happen to the coffee? It will get cold. Where does the heat go? To the environment. That's correct. Now, how does that affect entropy? It gets more dissolved because it's spread around more. That's correct. So, for example, another example is water freezing up. That decreases entropy. Why? أوجه ثانية واحد يجاوب واحد حماس. So when you freeze a molecule, the kinetic energy inside the particles themselves slow down and they shrink. So basically the entropy decreases or gets focused down to a point. So a good indicator of if something increases entropy or not, I'm not sure if you, can you see the highlighted point down there? Yes. So an exothermic process always increases the entropy, not always, but in the case, it always increases entropy. And an endothermic process always decreases entropy. Okay? So one of these following examples decrease entropy. Can someone tell me? 2. Okay, who answered? ياسر. هذول ثلاثة يلا هم ثلاثة جاوبوا من نفس الوقت. Her and you're not even looking. So okay. We go on to spontaneous process. Do you also get to explain what spontaneous processes are? So can someone explain it to me? No, someone else. Okay. So a spontaneous process means that under set conditions, it will happen naturally, without any enzymes, without any catalysis. It just happens on its own. It does not mean that a reaction happens instantly. So for example, an explosion happens instantly, but it's not spontaneous. Okay, so spontaneous reactions increase the entropy and release energy, so they're usually exothermic. For example, a rock falls is a spontaneous reaction, but it never falls upwards, for example. Or when iron rusts, that's a spontaneous reaction, but it cannot reverse back. For another example, ice melts below at room temperature, but it does not freeze at room temperature. Okay, so here's a question. Which one of these is a spontaneous process? Can you read? Okay. Okay. So D is leaving ice to I believe at room temperature. I misspelled it. C. Okay, so yeah, it's C. But I don't know who answered the first. When you read lollipop. Friday. No one got it. Okay. Oh, خلاص. Okay, can you tell me why it's a football rolling uphill? It needs energy. You need to put work in for the football to roll uphill. The rest you can leave them on their own and it will happen. Okay. So spontaneous reactions can be predicted with entropy. As I said before, when entropy increases, that means it's usually a good indicator that the reaction is spontaneous. Okay. When entropy decreases, that usually means that the reaction will not occur. And if entropy remains the same, neither process will occur. This is talking about reversible reactions. Okay. When the entropy decreases or entropy is negative, the opposite reaction will happen, which in that case, the entropy will be positive. Okay, so we move on to Gibbs free energy. This is 4.2. Does anyone know what Gibbs free energy is? Okay. So free energy is the energy available to do work. That's what it's defined as. It's given by this reaction, delta G equal delta H minus T delta S. So can someone tell me what delta H means? I explained it. Enthalpy change. That's correct. No. Yeah, enthalpy change. And T, temperature. And delta S, entropy. Okay. So it's used to predict if a reaction is spontaneous or not. When delta G is negative, that means that reaction is usually spontaneous because that means that the system has energy to give. Okay, and when delta G is positive, that means it has no energy to give and it requires energy to form the reaction. And when delta G is 0, it usually means that reaction is at equilibrium. Okay, so the following examples are delta G positive, except correct. Why is it hydrolysis of ATP? It would be negative. It will be spontaneous. Why? Because first of all, when you break down ATP, you'll form two products. ADP and organic phosphate. So when you usually whenever the amount of products are greater than the amount of reactants, the reaction is spontaneous and entropy increases. As you can see, the rest of the options were all catabolism. Catabolism, right? Or no, anabolism. So it's when things are forming. So that requires usually energy. You take it later. So here's equilibrium constant. It's good to know. So you can see here a reversible reaction with the products, with the reactants being A and B, and the capital A and capital B, and the products being capital C and capital B. Okay. Why is both the same thing? I don't know. It's supposed to be D. So as we see, when we use products over reactants with the power to the amount of coefficients. So for example, if I wrote to you, H2O will hydrolyze into 2H plus O2 plus O. How would I write the equilibrium constant for this one? EEQ equal. What will I put in the top? O H squared and H2 on the bottom. Okay, so you all have a general understanding of what equilibrium constant is. You'll not be tested for this, just to explain the rest of the slides. Okay. Listen. No. Okay. So here's Gibbs free energy continued. We can find at each side of the equation, if in a reversible reaction, if it's favored or not based on the Gibbs free energy. So when Gibbs free energy is greater than 0, which means that it's positive, what does that mean? It's non-spontaneous. And that means that it favors the reactants. So usually the reaction won't happen in the first place. Now when it's equal to 0, it means that both are equilibrium. And when it's greater and it's less than 0, which means that's negative. So it's spontaneous and that will favor the product. Any questions? No. Okay. So scientists studied the reaction below and found out that the products are present in greater amounts than the reactants. What can be deduced from their findings? 1. Why is it 1? It's spontaneous. So Gibbs free energy will be negative if the products are greater than the reactants. That means that favors the product side, and that usually it means that the reaction is spontaneous. Okay. Okay. So standard conditions. Have you heard about standard conditions before? You're supposed to learn it in the labs. So for example, when you enter a lab, what's usually the room temperature? 25 degrees, 298 Kelvin. Okay. And what's the atmospheric pressure? 1. Okay. And what's the concentrations if you're using certainly 1. Okay. So here's an exception. Here's an exception for biological situations. The pH will be 7. That means the H plus concentration is not at 1. So standard conditions are denoted by this circle thing. I don't know. And standard free energy is denoted by delta G, a circle, and is used to predict how reactions might occur. So in biological conditions as well, we'll have the apostrophe or the prime to denote that the standard free energy is in biological standards. So how do you use standard energy? Coupled reactions. So for example, if we have an endergonic reaction, which means that's endothermic and is non-spontaneous, how will I kickstart the reaction? I will use an exothermic reaction such as the hydrolysis of ATP to start to form the complete reaction. So what usually happens is that we have reaction 1, which is endothermic and non-spontaneous. We will couple it with reaction 2, which is exothermic, and that will form a complete reaction that is usually exothermic and spontaneous. What do we use usually for energy? ATP. So usually when we're doing exothermic reaction, coupling reactions, we'll use ATP. So the hydrophobic effect. Have you ever noticed when you're putting oil on or fat in water, when you're trying to wash a pan, that it doesn't mix? Why is that? Because it's hydrophobic. Now hydrophobic reactions, usually with water, I'll need to explode please. Yes, sir. Can tilt the camera? Okay. Tilt the. Okay. So usually when we have oil droplets in water, what will happen is that they will aggregate. Why? Because if we have, say, this is fat, if we have multiple fat droplets in water, what will happen is that we have to cage around or form a water, what is it called? OH and O bonds, H2O bonds around each of these fat droplets. So that will usually means that it constrict or tighten around the space that the water has to move around. So it will decrease in entropy. Usually when a decrease in entropy, what does that mean? The reaction is non-spontaneous, there won't be a reaction. So that's unfavorable for the water. What will happen instead is that all of these fat droplets will aggregate to take up the least amount of surface area to reduce the amount of water you needed to surround the oil. That will usually free up more space and will give around more entropy. So make the reaction spontaneous. Okay. Tilt it back. Now we move on to 4.3. So energy currency. Usually when we talk about energy currency, what are we talking about? ATP. Now there are two processes to power metabolic processes in your book. Can someone explain it? Okay. Phosphorylation and redox. So one phosphorylation and second one is redox. Yasser. أقول عبيدة. وصل البن. ما تعرف تلزم أنت؟ استغفر الله العظيم. Okay. So phosphorylation reactions. It involves the binding of inorganic phosphate to and off of molecules. So an example of these reactive molecules is ATP. So it is used to power any reaction, used as a token to power any reaction, so as currency, as money. And it is the most immediate molecule used for energy. So moving on to redox reactions. So redox reactions use the transfer of electrons from a more reduced molecule to a more oxidized molecule. These electrons usually carry energy, and there are three principal carriers in your body. Different face, please. FADH. Start with NADH. NADH. Oh, sorry. So I was explaining the electron carriers. So you know a rally race. So when someone passes a button to the next in a race, someone starts running. Okay. So that was the example of electron or redox reactions are. Now can someone please give me again the carriers? NADH, FADH2. We have also NADPH. Okay. So ATP reactions. ATP can be used to fuel immense amounts of reactions, many reactions, such as biosynthesis of molecules. You also have the active transport of systems across membranes. Can someone give me an example of that? You took it in biology. Sodium potassium pumps. That's correct. And then you also have mechanical work such as muscle contractions. Okay. So ATP is a nucleotide composed of adenine, ribose, and a triphosphate unit. It has two terminal phosphoryl groups, PO3, and are linked by phosphoanhydrate bonds. So ATP can be hydrolyzed into two reactions. Usually it will be hydrolyzed into the first one, which is ADP and inorganic phosphate. But when we really need energy, it's phosphorylated to AMP and PPI. And phosphoryl inorganic phosphate. Anyway. Pyrophosphate, yeah. So the role of ATP in metabolism, it's used as cash. So you usually have it immediately available. It's the most immediately available energy source and can be transferred from one reaction to another. So what would happen is that we would have, for example, glycolysis. We're breaking down glucose, sugar, to gain energy. And we use the ATP gained from the glycolysis to fuel anabolic reactions, such as building of fatty acids, building of DNA, and building of proteins. Also, we can use it as an intermediate to carry phosphate groups. For example, in your glycolysis, the very first reaction is giving an ATP, giving a glucose its phosphate group to form glucose 6-phosphate. So this is called phosphoryl group transfer potential, where it usually takes phosphate from a higher phosphate transfer potential to a lower one. So it's used as an intermediate. Now, why do we use ATP? We have 4 main reasons. Can someone state them? Okay. So at pH 7, ATP carries multiple negative negative charges. Now, what do you know? When negative and negative charge stack against each other, what happens? They repel. That's correct. So what will happen is that we would usually want to eject one of the bonds so it can take the negative charge with it. So that's for example, a motivation for the ATP to break down. Now, the second thing is that products of ATP hydrolysis are much more stable and solvable than the ATP itself. So that's another reason for it to break down. It will release massive amounts of energy. Now, ATP products are again more dissolvable than ATP itself, meaning that water can shield the ions of the ATP products to give more space to form more ATP, to have more ATP to break down. And finally, ATP breaks down into multiple molecules, increasing entropy. That means it's spontaneous. So, and then finally, we have energy and reducing power. So, we have the 3 carriers that we talked about: NADPH, and FADH2. All of these are electron carriers found in different parts of the body. For example, NADH is used to capture energy, usually from glycolysis and fatty acid breakdown. And NADPH is used in synthesis. So, for example, in synthesis of fatty acids and proteins. And then finally, we have FADH2, which is found usually in the citric acid cycle and fatty acid degradation. Okay, that's it for chapter 4. Do you have any questions? Okay.